you all for this opportunity, and thank you, Dale, for such a nice introduction about me. And yeah, like Dale mentioned, I'm a postdoctoral fellow at the Vancouver Prostate Centre, and yeah, I'm from southern part of India, and I've been here for past almost 3 years now. So this is where I work. This is our institute is located right next to the Vancouver General Hospital. We closely work with clinicians and a lot of patients who come in there. And in this building, we have a small lab set up here. We have about 250 people working in with different parts, mostly on prostate cancer, but some people do work on other stuff also. There are a lot of we have a mixed bunch of people. Like we are clinicians, some basic scientists, and there are computational people, and there are people who model drugs and stuff together. And yeah, we have a very cohesive team, and it's amazing to work here. So from that, I just want to make sure that I put this out. I'm not a practicing physician, so whatever I'm describing here is from my research experience and my personal knowledge. So so if you need anything, if you find something which is not agreeing with you, please always discuss with your physician. He or she will have the best thing for you, and in terms of whatever with you or anybody you know regarding the disease and how the treatment is done. Said that, I do want to begin with, as I said, my topic is about finding new therapies in prostate cancer. So when we speak of prostate cancer and the therapies, it all starts with Dr. Charles Higgins, a Canadian fellow who in late 1961 first showed that when you castrate mouse, which had dogs and mouse which had prostate cancer, the tumor volume actually shrunk. So this is what got him the Nobel Prize in 1966, and from then the removal of testosterone or the castration has remained the primary state of treatment of hormone responsive prostate cancer. And from the past 70 years, we have been working on this, trying to understand, but more coming, develop better therapies, but all in all against in terms of castration itself. We haven't really ventured out too much. We have a lot of new therapies coming in, but this is and has remained the main focus of understanding prostate cancer growth. So because of this, it's extremely important to understand how this works. So testosterone, the dihydrotestosterone, which is the active form of testosterone, they together constitute the androgens in human body. So androgens are what makes the male part of like what you describe as the manliness, and so-called quote unquote is all derived from these testosterone. Like either you're growing long beards or bulking up the muscles, they are all kind of derived and mediated by the testosterone. While these are amazing characteristics you want in your body, this also acts as a fuel for the development of prostate cancer. Basically, while testosterone is important for the development of secondary sexual characters and as well as growth of your prostate per se, but in terms of a cancer, it kind of adds fuel to the fire. So our aim so far has always been to kind of put out this fire or take out the fuel, and so that the growth of the prostate is going down. So while I was mentioning about testosterone, it's also important to understand how this testosterone actually works. So this androgen, which is now so I'm showing the picture of actual one cell here, a prostate cell, which, so once the androgen is present in the blood, what happens is it comes and binds to this receptor, one protein in the cell, which recognizes this dihydrotestosterone, and it comes and binds to this. So once it binds to this moves from the cytoplasm of the cell into the nucleus. So it's all the magic happens in the nucleus. And androgen receptor belongs to one kind of family of proteins which we call as transcription factors. Transcription factors are nothing but the ones which dictate which gene to go up, which gene to go down. For example, so if you eat sugar, so your body will send a message to make more insulin. So the insulin gene is turned on. Then the insulin gene, what makes is an insulin protein, which kind of, helps in metabolizing your sugar, and your sugar level goes down. So like in terms of diabetes, when there is no insulin production, your sugar level keeps up. So it's the same thing which is happening. So everything is at the bottom level is what is controlled by the expression of the gene. And androgen receptor also does the same kind of thing. It activates certain genes which are important for the prostate cancer growth. So when you take out the androgen receptor, these genes won't go up, and the growth of your prostate cancer will reduce. That's the whole idea in a very simple way. And we have been trying to understand, like my whole PhD was understanding how this works. Even though people have been studying it from past 60 years, there is so much more to understand about this. And I was learning a bit small part of it during my PhD, and I'm continuing a kind of different angle, but in and around the same area. So like as mentioned, so anything to do, so our whole aim is to identify mechanisms, how to stop this from happening. So over the years, what we have understood is androgen receptor and is the central for growth of almost all kind of prostate cancer, whether it's benign, it's responsive to hormone, or late stage metastatic cancer, all have androgen receptor, and it occupies the central role in the development of prostate cancer. So naturally, this is the mainstay of treatment in treating prostate cancer even today. So the idea is to stop this androgen receptor. So the two things which we usually do is either stopping the production of androgen receptor or inhibiting the androgen receptor from doing its work. So for the production of androgen receptor, what we use is something called as LHRH. So luteinizing hormone releasing hormone is a small hormone which is released from the pituitary gland, which is located in your brain. So what this does is it sends out a signal to your testes to make androgens. So the idea is you're either by giving an agonist or an antagonist. Agonist is something which make it so much that it kind of loops back saying that, oh, there's too much, let's stop making it. Or antagonist is something which blocks it. So what happens is when this LHRH hormone is not there, it stops, it prevents the testes from making more androgen. So the idea is if there is less androgen, the growth of the prostate, like because it is the kind of which drives it, reduces it. So the second thing is an enzyme which is required for the conversion of testosterone to dihydrotestosterone, because dihydrotestosterone is the active one, and it is what it makes the prostate to grow faster. So the other thing which we do is you inhibit this enzyme and preventing it from making the active testosterone, which is important. After this, the second type is direct inhibition of androgen receptor. So like I mentioned in this earlier figure, which I showed you, so this androgen has to bind to this androgen receptor in order to do the remaining things. But when you have an anti-androgen, which kind of binds to this receptor, it prevents the binding of androgen. So once this doesn't happen, whatever downstream was supposed to happen doesn't happen. So these are the two basic ideas which we have been dealing with so far, and there have been many new drugs, either from preventing synthesizing of more anti-androgens or developing newer anti-androgen therapies. And we have been pretty successful and has been used as standard of care now. But so once, like if you actually plot out the growth of the tumor versus time, you see there is, this is during the initial diagnosis, you see a growth in the tumor. This is when you realize, okay, there is something wrong. You get your PSA checked, then you get your, the physical therapy and all those things. And the doctor probably decides if they're responsive to hormone, they decide to give you androgen therapy, anti-androgen therapy. So it works extremely well, and there is a regression. Immediately, you can see there is a reduction in the tumor size, and most of the people are extremely happy with this, and they're going by fine. But there is always some people who at a later stage kind of have a relapse with this. And these, at this time, when this tumor comes back, they become irresponsive to this anti-androgen therapy. So this is what we call as castration-resistant growth. So these tumor cells, whatever probably was remaining, they kind of get develop a mechanism which can be from so many different things. They learn how to grow without the testosterone. So this kind of acts as a problem for us, because now we can't really give anti-androgen therapy. So we have to give something else to make them stop. So from past few years, what I have been studying is identifying new modalities to treat these castration-resistant prostate cancer. Like what kind of new therapies we can identify to kind of reduce the growth of this castration prostate cancer, to make people's life more better. This has been the work which I've been doing. So I will focus on two different things which kind of I have been dealing with in the last last few years. So one of the things was work which I was kind of given the scholarship from the PCFBC to support this work, and the other thing which I'm currently doing right now in the lab. So there are so many options of going from here, and the two things which I'll focus on is something called as personalized medicine. The other thing is identifying new therapeutic targets. So I'm sure you guys must have heard about personalized medicine, but let me there are so many ways people can go about in identifying therapies which are very personalized to certain population or certain person. So one thing which has been pretty common these days is something called as whole genome sequencing. So not so common yet in everyday clinical practice, but it will be, and it is going. It definitely, considered from past 5 years, it has become more common. Like routinely, a lot of doctors prescribe doing, if not the whole genome sequencing, at least some part of sequencing to identify what is actually happening molecularly, what is different, so that they can better decide to give a drug which kind of matches you, because we are all so different. So whatever we have is also different, right? So like something which works for person A may not work for person B. And this is something which we have seen more and more in cancer compared to other infectious diseases. So we need something which demarcates you, or which in each cell, what defines you versus somebody else. So one thing which we focus is something called as mutations. So if you are not very familiar with mutations is, if you imagine, like our DNA has a lot of repetitions of the same code, which is ATGC, and it's been mumbled and it has been written so much. So if you can imagine, like an Encyclopedia Britannica, for example, and if you ask somebody to keep copying it every day for millions of times, they are bound to make mistakes. And that's what really happens in our body too. When it keeps on copying this, there are few mistakes which make. So even though our cells has a backup plan to correct it, some mistakes go, kind of misses that, and they become prominent. Depending where these mutations are, they can be problematic. So for example, if the mutation is an important gene, so what happens is that gene might not function. So for example, there's something called as tumor suppressor genes in our body. The main function of these genes are to suppress the growth of these kind of mishaps from happening. But if there's a mutation in these genes and these genes don't work properly, so the chances of somebody getting a kind of, those cells behaving differently increases. So in mutations, there are basically two kinds of mutations. One is something called as germline mutations, which are inherent to you, like it has been passed on from our parents and stuff like that. I'm sure you recognize this from the case which was kind of very popular, for which Angelina Jolie got mastectomy done preventively. So because she realized that there is a mutation in a gene called as BRCA2. So studies have shown that people who have the mutations in BRCA2 are more, the chances of them having breast cancer increases quite a lot. So she got a mastectomy done. So while this is germline mutations, and there are few which have been identified and people are routinely screened, there are something called as somatic mutations. These are something which you acquire over the period of your life. And these mutations sometimes can be in the gene, and many of the times they are not in the gene, but in the region next to the gene. So these regions are something which regulate the gene. So my interest is in understanding what kind of mutations are there, and how we can use them to identify personalized medicine. So the problem, one of the biggest problem with this is when you do a whole genome sequencing, there are thousands of genes, which mutations which are identified. It is impossible to go and identify or validate or test each of these mutation. So we had to come up with a solution to at least narrow down, divide a pathway or a kind of a pipeline where we can take these hundreds and thousands of mutations which we identified from whole genome sequencing and come up with a way which actually can identify actionable mutations so that we can test them and figure out if we can use them for further clinical applications. And that's what we did. From if you have to identify one mutation which is important, or a few mutations which we want to test further from this hundreds and hundreds of thousands of genes which we identify, is a monumental task. So we came up with something called as a methodology called Starsign, though I don't want to really go into telling you how it works. So we developed this methodology based on assumption or hypothesis that the androgen receptor, wherever it binds, these regions are very important, and we can functionally test this, which are important. And based on this methodology, we kind of identified this roadmap to narrow down thousands of mutations to important mutations, which are in 10 to 20, which we can actually test in the lab very quickly and figure out if these are important. And this was the work which was funded was supported by Prostate Cancer Foundation, BC. And what we see here is using this methodology, we actually seen that of thousands of mutations, we actually identified a mutation very quickly, which is located in an important gene called ZBTB16. And we actually showed that when this mutation is actually important, and when you kind of manipulate it, the expression of the gene goes down. So this is just a proof of principle, though this is not really telling you, we are not identifying anything downstream, but this is just a proof of principle saying that the methodology which we kind of worked on, or the roadmap which we have done now, is actually important and it can actually work. So just like me, hundreds and thousands of researchers who are trying to work on this can now use this platform to actually come down and identify few tens of genes. So if everybody tests few tens or 20 mutations, in a quick time, we can identify many important mutations which can further be used for clinical application. So this was one thing which we recently worked on when we actually wrote a paper on this, and it got published also. This is one part. Second part, which I'm now currently working on, is identifying new therapeutic targets. So while androgen receptor and targeting this has been so important, and we have been doing it for so long, like I said, it kind of is hitting a saturation, and we want something alternative to target on. So I want to take back to you on this original picture, because this is kind of the most important thing about understanding of prostate cancer. So while I showed that androgen receptor sits on the DNA and kind of controls the gene expression, it doesn't do on its own. What it does, it does it in a team of different proteins, what we call as co-regulatory proteins. There are a bunch of other proteins which help to do it. And they're also very important in telling how the function of androgen receptor works. So instead of directly targeting androgen receptor, we can actually target any of these so-called co-regulatory factors, or the proteins which are helping them, and kind of change how the whole system works. And that is what we have been working on. Based on this, we have purified all the AR co-regulatory proteins using a proteomics approach, and we have identified one of the important protein which affects which works with androgen receptor, and it is called HOXB13. And we have in the last few years have shown that it is very important for prostate cancer growth. So we did some initial experimentation to see how it affects the growth. Everything is in now in petri dishes or in a mouse or stuff like that, though we haven't really taken it to the clinical aspect. But our initial experimentation shows that HOXB13 is only present in prostate cancer, which is a very added advantage for us, so that if you kind of try to inhibit this, no other organ will be affected, but only prostate. So what we saw was when we took some prostate cancer cells, we took out this HOXB13 and injected into the mouse. So the left side is a normal cell line prostate cells which do not which have HOXB13, and the right hand side is where you don't have HOXB13. So, and you can see there's actually no tumor which is growing on the right hand side, but in the left hand side there's a big tumor which grows, which is very our preliminary results shows that this is something which is very interesting, and kind of we can pursue it further to devise a mechanism how we can inhibit, like maybe we can get a drug or some other mechanism to inhibit HOXB13, and this can be a viable option to treat patients further. And that's what we have been working on right now. So one important part of developing a drug against HOXB13 is actually identifying its structure. So we are now purifying the protein and identifying how it looks, so that we have to develop a drug which exactly matches it and sits to understand and kind of sits to it and inhibit the function. And that's what we have been doing from past couple of years now. And the other thing is we are also working on understanding how HOXB13 per se works in our body, and what are its functional roles. Yeah, this is the ongoing work. We are very optimistic of our results, though bringing it to a clinical practice will not be an easy job, or it won't happen in a day. But we are optimistic of the results, and hopefully in near future we will be able to bring something out into the market. That's all. Thank you.